So today we're going to look at two things. One is the pentose phosphate pathway, which is a way to make a bunch of NADPH and a lot of precursors for DNA, RNA, and other things. And then the second thing we haven't looked at yet, but we will, is how does actually glucose enter the cells, and what happens in type 1 diabetes. Okay, so how is the metabolism get affected when someone has type 1 diabetes? So those are the two things we're going to look at. Okay, so first thing is going to be the pentose phosphate pathway. So this is what you start with G6P. What is that? We know what that is, right? Glucose 6 phosphate. So G6P will go here to this thing. There are a couple of steps to go to this compound. It's called 6-phosphogluconate. This is actually a step where we oxidize it, and so this is the enzyme is going to be another dehydrogenase, G6P dehydrogenase. So it's a dehydrogenase, so we know we need what? Either NADH, NADPH, or NADP plus or NAD plus. We don't know yet which one we're going to need in this case. So normal dehydrogenase, that's what they will use. So this is an oxidation step, right? So now tell me what you need. Which coenzyme do we need? Oxidation. NAD plus. So it turns out in this case we don't use NAD plus, we use NADP plus. And what are we going to generate? So do not confuse NADH and NADPH. They really serve different roles. Okay, their metabolic need, the purpose for these coenzymes is quite different, right? So we're going to see what that is. Okay, we make NADPH, and whenever there is NADH or NADPH, we need to have the proton. So similarly, when you have NADPH, we need to have the proton. So we made 6-phosphogluconate, then there's another oxidation state here, step, and we make another sugar. It's called ribulose 5-phosphate. This is also an oxidation state as we release CO2. And here also we are using NADP plus to make NADPH. And this ribulose 5-phosphate will go on to make ribose 5-phosphate. Go on to make DNA, RNA, and there are some coenzymes. Glucose that enter the cell goes to G6P. We know we go to G6P from glucose, right? Which enzyme? Kinase, right? It's a kinase. But G6P, see that now the first time we're seeing G6P does not only go to glycolysis. It can also be used to make DNA, RNA, and other coenzymes. This is called the oxidative phase, where you're actually oxidizing G6P to ribose 5-phosphate, right? Now we're getting a bunch of NADPH. So this is one of the major pathways by which we get NADPH. The NADPH production is really vital for a lot of metabolic processes. Okay, so that's what we're going to discuss next. So now you can tell me whether NADPH is going to be a reducing agent or an oxidizing agent. It gets oxidized. So it is a reducing agent, right? It must give electrons to something else, then it gets oxidized. just similar to NADH. There is, it's just very similar chemistry. This also is the hydride donor, right? NADPH will be hydride donor to go to NADP plus. So NADPH is going to be reducing agent. So in our case, what happens is that this NADPH actually reduces glutathione to GSH, and the enzyme here is going to be glutathione reductase. Okay, so what that does is that will reduce oxidized glutathione to go back to its reduced state. And again, in a minute, I will explain what that is. So the other purpose of using NADPH is to actually provide the electrons during anabolic processes. So we are using precursors to make fatty acids and sterols, and this is part of anabolism. And so this is also reductive, right? We're reducing things to make new compounds. So now, once you use NADPH, what are we going to produce? What happens to it? It goes back to NADP plus. So we are regenerating NADP plus this way so that it can go back here, right, to make the other steps happen. So this is a redox scavenger. What does that mean? So I will not draw the structure of glutathione, but you remember cysteine? Yeah. So what is special about cysteine? Disulfide, right? It can be oxidized to make disulfide. So glutathione also has an SH, CH2SH, that can be oxidized to make disulfide. So this is your glutathione, and it has that SH. And so when you use two of them, then we make oxidized glutathione, right? S, and that's our disulfide bond. And so if this is GSH, this will be your GSSG, right? Okay. So that is what GSSG, that's your oxidized glutathione. And the role of glutathione is to actually protect us from harmful oxidative damage. So what happens is if you have hydrogen peroxide, for example, then you can make water, but hydrogen peroxide is actually not good for us. It also generates these radicals, hydroxyl radicals, right? And what they can do is they can damage, so they can damage your biomolecules. So DNA, RNA, everything, like I don't even have to write. Anything that comes in their path, they will oxidize, right? This is highly reactive radicals. And so normally, like all of us have some reactive oxygen species. This is why we eat fruits, right? Because they have a lot of antioxidants, right? So antioxidants, what they do is they will neutralize this reactive harmful species, right? So our body is protected. Make sense? So this will be called reactive oxygen species. what glutathione does is it scavenges. Glutathione scavenges this reactive oxygen species to make water and itself be oxidized to oxidize glutathione. So glutathione scavenges this reactive oxygen species to protect us from oxidative damage. Remember, if you oxidize it, then it cannot protect us, right? Do you see here? We need to have it in the reduced form so that it can scavenge the reactive oxygen species. But once you oxidize it, then it doesn't have the protecting form anymore. So how do we solve that problem? We reduce it back, right? And how do we reduce it? So we use NADPH to reduce it back. So if we do not have, you see the big picture now, if we do not have NADPH, then we will be attacked by reactive oxygen species because there is no glutathione to protect us. So we have G6P, right? And this is our normal, this is our pentose phosphate pathway. We go this way. And the purpose here is that we are making NADPH, which is reducing agent. And part of its function is to reduce the oxidized glutathione back to its reduced form so that this GSH can help us maintain our help our bodies to protect from reactive oxygen species damage, right? Go ahead, Natalie. NADP plus. Right. That's right. Yep. Yep. So yes, so when we reduce the glutathione back to its reduced form, we regenerate NADP plus, which can be used here, as well as when we make fatty acids, we also make NADP plus so that they both can contribute to this pool of NADP plus. Go ahead. So how many electrons? So cysteine, so just think about cysteine. When you oxidize cysteines, right, to disulfide bond, we know it's redox process. And well, I already wrote here, it's a 2 electron, 2 proton process. So we need 2 electrons, so the same chemistry. We need 2 electrons. So now you can tell me whether you need one NADPH or 2 NADPH. So NADPH, NADH, they are doing both the same 2 electron chemistry, but their purpose is very different. They can both act as reducing agent. NADH is not used to make fatty acids or in this pathway of reduced glutathione, only NADPH is. NADH has other roles. Go ahead. Okay, so the question is whether NADPH is an oxidizing agent or reducing agent. Hmm? So how do you define reducing agent or oxidizing agent? So reducing agent is something reducing something else. If I am reducing agent, I am reducing something else. So I must give electrons, right? Then what is happening to me? I am becoming oxidized. So reducing agent is something that reduces something else, itself being oxidized. So I'm going to write a few full sentences, okay, to sort of summarize this thing. So redox homeostasis. So redox homeostasis means to maintain the balance, redox balance within us. So we have a lot more NADPH than NADP plus, right? What about here on the top? we have a lot more NAD plus than NADH. So now you need to tell me what that means. The numbers make sense, but broader, bigger picture, what does it mean? Yes. So that means we have high amount of NADPH because we need that as a as a reducing agent because of these things, right? But NADH is very low. Between these pairs, this pair, top pair, is used mostly for oxidative purposes, right? Because we make NAD plus, we use NAD plus. But for reductive purposes, NADPH is what is our primary coenzyme. So you look at regulation of G6P dehydrogenase, right? That's the enzyme that made NADPH and oxidized G6P. We can see that G6P is partitioned between glycolysis versus PPP, right? So how do you know which way to go? We're using one precursor. We can either do glycolysis or we can do PPP. So how do you decide which way to go? And preferentially, which one to promote, which one to inhibit? So NADPH is a feedback inhibitor for G6P dehydrogenase. What it means, if we have a high enough NADPH concentration, then this NADPH is going to inhibit G6P dehydrogenase. To stop pentose phosphate pathway, but promote glycolysis. So it means that if you are not consuming NADPH at a rate at which it is being made, then there's an excess of NADPH, right? So we are not using NADPH to either maintain redox homeostasis or we are not making fatty acids, for example. Then why make more NADPH? Right? Instead, use G6P to do glycolysis, or it is also used to, not only glycolysis, but you can also use it to store glucose as glycogen. So do those processes instead of going this way. So that's what it means.